Okay, so we are here on a Tuesday in July for the second of our Summer of Symbiosis book club discussions. And today we've got a bit, I think people are off doing fun summary things or off trying to desperately get done as much research as humanly possible in the summer. So Mark and I are here, and we are missing one of our normal stalwart companions, Karen, who is working hard on doing her finishing up with her PhD. So hopefully she'll be able to join us for our last one in August. But we give her a little shout out here, and hopefully she's doing well. Mark?
She's our third microbial musketeer, and she's much missed.
That's right, exactly. Yes, that is an excellent way to put it. So if you stumble across this while we're actually doing this live, then feel free to pop in. We have the link posted at our website, which is g+microjc.wordpress.com. There should be I should have something posted right here that has things written out so that people who, you can actually see it. I haven't figured out quite how to do that yet. So I am Laura Williams. I am a new assistant professor at Providence College, and I'm working on, my research focus is on symbiosis and bacterial lifestyle, but I'm also teaching a course in the fall for the first time on symbiosis for our biology majors. So this is kind of my selfish excuse to read a lot of symbiosis work so that I'm getting ready for my course in the fall. So I'm excited about the book we're going to read today, or we did read and we're going to talk about today, which is The Symbiotic Habit by Angela Douglas. And then I will let Mark introduce himself.
Hi, I'm Mark Martin, an associate professor in the biology department at the University of Puget Sound. I love me most matters microbial, it's true, and I've worked on mostly undomesticated microbes throughout my entire career. I love predatory microbes, I love symbioses. As for myself, I taught a freshman writing seminar dealing with parasitism and symbiosis. Shh. One's kind of a subset of the other, but the students don't know that yet. Definitions are important. I too thought this book was fabulous, and so I'm delighted to have this opportunity to talk about it with folks.
Yeah, me too. I am really glad to have an excuse to have read this, because I think that I'd seen it and I'd not committed to reading it, and I'm really glad that that you suggested it. So this was your suggestion, and I'm really happy for it, because I think there's a lot that's packed in there. And Angela Douglas, I met her once because she is an insect endosymbiont person, amongst many things. And I did research on insect endosymbionts in my postdoc with Jen Werner-Green. And so I met her once, and she's just fabulous. And so I'm really glad to have gotten a chance to sit down with book-length work that researchers put out, because it's so hard to get all of these conceptual things and thoughts that you have about a broad take on the field. It's hard to get that into a paper. So it's nice to be able to read somebody's thoughts on that in book form.
And it's very frustrating, because this is a wonderful, wonderful book published in 2010. And when you think about all the changes since then, I mean, how tired you must be. I remember when I was trying to come up with things for students to read, I too wrote to Professor Douglas, and I can't really make this plain, but there was almost a weary tone in her email. Yes, I'm working on another one, because I don't think you can ever catch up. There are always new changes. And so I don't know, Laura, what your clientele for your course is going to be. Do you know yet?
They are mainly going to be upper level biology majors. So I think this is definitely going to help me with the course, but I'm not sure if I'll be assigning and reading from this.
Yeah, I have a little bit of a different problem in that, my job is to kind of work with incoming students from a variety of backgrounds. I would say roughly half are science majors.
Mm-hmm.
They're all real bright, they all real enjoy it, but it's hard to get them up to speed. And if you look at, this is not a thick book, but it is a very dense book.
Well, it's really interesting too, because I think it's about the same length page-wise as the first book we read, which was 1 + 1 = 1. Uh-huh. But the style and the intended audience are very different. So John Archibald's book was aimed, I think, at the general public. The first part of the book, if you were a scientist that knew anything about molecular biology, you could get through it pretty quickly. This book is just like Symbiosis. Let's just go there. And then you're like, okay, all right, here we are.
The fire hose, exactly.
Yes.
It very much was like that. But what I would say, and I don't know if you, many people don't read the introduction or preface, I should say. And one thing that really struck me here is dividing here into the 3 things that are most important. And you know, molecular and genomic techniques, good Lord, you've been part of that, Laura, and the changes that have taken place. To me, number 2 is almost as important, conceptual, the way that things changed. For example, the term mutualism in general.
Yes.
Finally, relevance to the everyday world in ways perhaps trying to heal coral reefs or looking at how to improve health issues, because I think that this issue of symbiosis is critical, especially since my whole family became kind of intestinally ill on our last trip. I've been doing much thinking about dysbiosis here on a personal nature. And so for that reason, this whole idea of the interactions between everything are a lot more than thinking of a hermit crab with a sea anemone on its shell. I mean, everything's interacting. Well,
and I think that actually leads into, I think, the main thing that I wanted to start with and get your perspective on, especially because I'm sure you've got some thoughts on this. And this is going to be relevant for me for teaching. And you just said, you know, definitions are important. And so she starts out, and I love what, so she starts out very, very logically with defining what her definition of symbiosis is going to be for the purposes of this book. And one of the things that she says is, I'm trying to find the quote. The term symbiosis was coined originally by Anton de Berry in 1879 to mean any association between different species, with the implication that the organisms are in persistent contact, but that the relationship need not be advantageous to all the participants. She says many symbiosis researchers use this definition, and without doubt, some colleagues steeped in the symbiosis literature will have objected to the opening 2 sentences of this book, which are, individuals of different species form persistent associations from which they all benefit. These relationships are symbioses. So within the first 4 pages, you get the sense that this is a term that encompasses a lot of what we understand about interactions between organisms, how ecosystems are founded, how organisms can make it in different environments, but we can't particularly agree on what's meant by this term symbiosis.
Exactly so, and you'll notice even when she gives her new one, the working definition. And I love this. This is one of those understatement sentences I like so much. I subscribe to this definition, even though it is not without difficulties. Yes.
Yeah. What page is that? Is that the? Oh, there it is. It's on 6. Yeah, the top of 6. Yeah. And so one of the things that I returned to as I was reading the book, she starts to kind of draw, and with any definition you have to kind of start to draw, you have to kind of circumscribe, like, what are you thinking, what belongs in the circle that you're calling symbiosis, and what have you by then the definition excluded from outside of that. But it's a porous circle instead of, you know, a strong barrier, because she says, oh well, there's, you know, like pollinators and their relationships with plants. Right? That's probably not a symbiosis, but we're going to talk about them some because they have some helpful parallels with what. So you kind of, it's this like shifting, it's a shifting definition, and it ends up encompassing some things but not other things. And is it always a mutualism? What do we mean by mutualism?
What do we mean by it? Yeah. And I mean, if you think about it, the hand of Darwin is on most living things. So you look at pollinators, and there's clearly selection on both sides of that, even though there isn't, quote, a symbiosis in the stronger sense. And what I'm concerned about a little bit, and I'm not being negative at all, is that people come in with very strong ideas of what the term means. For example, the vast majority, you can check this with your students, find symbiosis to be a positive term, not a negative term. And in fact, there's nothing about the term that's positive.
Yeah, unless you choose to kind of enforce that requirement on it. Yeah.
And this is the big thing that we want to deal with as scientists, the issue that one size doesn't fit all, right? Versus on the other side, trying to be very precise in the way that we define things.
So I'm curious. So there's kind of, when I keep running across people's definitions of symbiosis, there seems to be kind of two aspects to it that people define in different degrees to try to say this is a symbiosis. And one is the length of contact between the partners. You know, on the extremes, you could say, are they like with insect endosymbionts? Those two partners exist together, one inside the cells of the other. They are always in intimate physical contact. And on the other extreme, you have things like, if people would suggest like pollinators, where these two species have co-evolved, but you have one coming in and then there's a brief interaction that's incredibly important for both, but then that interaction is broken off.
Right.
So those are the two extremes. And then the other kind of part of the definition seems to be, what are the consequences or benefits to the two partners? So for something like predatory bacteria, which we are both, you know, interested in, it is a predation of one bacterium on another, and then it's a contact that ends with one partner dying and the other one going off. So would you call that a symbiosis? It depends on your definition, but would you put that into the realm of symbiosis?
And this takes you to the Mortimer Starr PDF, and I believe I sent that to you from, this is the one where he goes on for 20 some odd pages on the definition. Parasite, parasitoid, symbiont. It's a very difficult thing because classically symbiosis does not take into account the nature of the association in a positive sense. You get that, not a Venn diagram, but the, you know, the A plus B minus, you know, the way that the partners associate with one another, and that's classically how it's taught. And I like that on the one hand because it shows some varieties, but when you talk about things like, oh, see, I'm so mutualistically minded because Margaret McFall would hit me if I didn't say that. What's the other one close to that? Where's my brain for that? And it sounds like it would be positive.
Sorry?
The other association that's supposed to be, where one member is neutral.
Oh, commensalism?
Commensalism sounds as if both members benefit, right?
Yeah.
And I love talking about that with students because they say, how can it not have an effect on the other organism?
Yeah, absolutely.
In terms of space, in terms of energy resources. And I think that's where she comes to in this book a lot, is talking about energy partitioning. What you're talking about is a very interesting concept. If they're not in intimate contact, what do you say? Mm-hmm. And this, as far as predatory bacteria go, the predatory microbes really surprised me because they had much larger genomes than I expected, right? Mm-hmm. And all the ones that we've looked at so far don't seem to have particularly reduced genomes, as you might expect. Mm-hmm. And that suggests to me that it might be an alternative lifestyle. It reminds me a little bit about insectivorous plants. They don't actually need to eat insects, right?
Yeah.
You probably saw Young had written about the nepenthes. That's the plant that encourages bats to come poop on them for fertilizer. You know?
Yeah.
So kinds of associations are very interesting, but I don't think we can get to the meat of things until there's that kind of intimate association.
Yeah, I think that's true too. And I think in my own, as I've kind of started to navigate, starting a couple of years ago, tried to kind of navigate developing my own independent research trajectory. So I was doing that within the context of working in a postdoc on an insect endosymbiont system. So I was thinking in terms of symbiosis. And I actually have a book that I got, I'm not going to remember the names of the authors. It's just called Symbiosis, and it came out, oh boy, it came out a few years ago, and it's just kind of like almost like a textbook. Somebody wrote a textbook on symbiosis. Two authors. Oh, here it is. Surinder Parasar and Vernon Ahmad. Sorry. And I got that book, and it included predatory bacteria in it. But they very much said that they're like, we are casting the broadest net possible at this point because we just want to talk about all of these associations. And so for me, I've been trying to kind of think very carefully about what are the terms that I want to be using as I'm describing.
Yeah.
What, and I think lifestyle, when you said lifestyle, I think that's a better way, in terms of the predatory bacteria system, I think that's a better way to think about it.
Well, here's something I'm glad you're here, and it is relevant to today's discussion, though it's not in the book, and I don't believe I've had this conversation with you. Years ago, Joe Handelsman's group had published on looking at how some bacteria can take advantage of peptidoglycan being shed by Bacillus cereus. And if you read the paper carefully, and I'll send it to you if you're interested, and I've asked lots of people about it, and I never get a really direct answer, so I think it's one of those projects people work on and never finish. It appears as if there are bacteria that live inside the peptidoglycan layer of Bacillus cereus. Yeah, that's the look I'm looking for on your face.
Really?
Yes.
I had not heard about this.
I call them peptidoglycan surfers. And I would love nothing more than looking at this association, because my guess is that they're not going to be able to live in other places efficiently. Now, there are many predators that can, and not Della vibrio, but there are other predators that can live on strict media that you have in the lab, and it's only under some circumstances like nutrient deprivation or some other signal that they exhibit predatory activity. But this idea of being almost to the case where you're assembled this way permanently is what's interesting to me.
Yeah.
And I've never, I see you're very excited now.
Yeah.
And I'm not exaggerating. It is in the paper. I will send it to you. It's from, I think, 2004, I want to say now, but I've asked about this plenty of times over the years.
So can you, I mean, because I've not heard this at all. Soil
isolates. They're soil isolates of Bacillus cereus, because Joe's lab used to work on Bacillus cereus because one species made a particular biological compound that inhibited a fungal disease of sprouts.
Okay.
They did that. And in the process of isolating these from the soil, they found out that even after 3 colony isolates, they would get these gliding bacteria co-isolating with them, even though they went through 3 single colony isolations.
Yeah.
And those gliders love them some peptidoglycan.
Interesting.
And my guess is they're surfing it, because in Bacillus cereus, you have lots and lots of layers, not just 2 or 3, but 50 or more.
Yeah.
And my guess is that's a whole environment to live in.
Yeah.
Yes, that idea. Look at you.
What happens when they form spores?
That's a wonderful question. Okay. You know how much I love that kind of thing.
Yeah.
And no one followed up on it. So, I'm to the point, because I've asked about it, and you know how much I am grateful to and adore the Handelsman lab. I don't know if anyone's working on it or there's just so much stuff going on, but I'm almost inclined to start looking in soil samples myself. And do you see things like this?
Yeah.
And the paper, in fact, we should probably post it so other people can look at it too.
Yeah. Oh, and I've said I'm going to post things before, and I never do, so I really will do it this time.
I send stuff. No. I try to be a good musketeer.
You are. I'm the like. What am I? I'm the one that's constantly just wandering off and not finishing missions.
I guess I'm the old drunk among those musketeers. There we go. Best line ever, right?
Yeah, that's really so. Okay, so that's yeah.
Again, what we're talking about, to get back to the point of the book, is the issue of it's best to study symbioses when there are these really strong associations where there's clearly resource partitioning and transfer.
Right.
Now, I was trained in graduate school to look at rhizobium and forming of root nodules. And as you recall, during the formation of a root nodule, you get differentiation of those bacteria into a form called bacteroids. And those bacteroids actually cannot grow very well on their own, but there are still within every nodule some that have not yet fully differentiated. So within those bacteroids, you think, so they're going to be importing all kinds of goodies. But it turns out that the membrane of the plant material segregates what metabolites are available to those bacteroids. So, my point on that is that it's really kind of a two-sided thing.
Yeah.
And that's the type of symbioses that I think the symbiotic habit is about.
Yeah, I agree. Yeah.
Now, you look at things like Riftia worms, and it is very clear that the bacterial symbiont disperses, but no one has ever found it growing anyplace.
Yeah.
In that sense, it reminds me of some of the vibrios that inhabit squid, that you can detect them in the water, but they clearly make a beeline for their partner.
Yeah. And so I think you're right. I think if you're going to be asking questions about how these relationships form and are maintained, and you really are looking at things that have an association probably, I mean, you're talking about physical contact between them. And that the duration of that is a little longer than just, for instance, something coming and pollinating and leaving again. So I think that's a good, and that's something I'm thinking about as I'm thinking about designing the course for the fall, is just how are we going to set up the parameters for what we're going to cover in the course. And
so I make a joke about it, Laura, where I often say that, variables aren't and constants don't. That's what I heard in physics as a joke. And so when you're teaching genetics, and symbiosis is very much like genetics in the sense that it's an extremely broad field where you have to know a little bit about a lot of different stuff, which is what's exciting about this book as well as what I do. I mean, I'm really a geneticist. And you got to bring together all these different ideas. And so when you talk about the establishment of the symbiosis, and you look, for example, at Buchnera and aphids, you have a fossil record, you have molecular data, this is a 50 million year old symbiosis, yet there are other examples that seem to happen very quickly, the X bacteria of geonic.
Mm-hmm.
And then you see figures sort of like this.
Yep.
Right, where it makes it appear as if the changes between being non-pathogenic or pro-symbiosis are in one or two genes changing, and I'm sure that's the case for some things.
Mm-hmm.
I don't know how you feel about this, but I think that most alpha proteobacters are kind of preset for associations with eukaryotic cells, because I'm trying to think of other types of things like that, and they're mostly alphas.
Yeah, like the rickettsia and so forth. Yeah, and I think that's one of the things that I've in this book was really helpful for this is to try to organize. If you're going to teach a course that's entitled Symbiosis, one of the things that you want to do is try to figure out what other concepts are you going to try to tie in that symbiotic systems are useful for exploring. So when there's parts in the book where it talks about the development of cheating in a partnership, in a symbiotic partnership, and when do cheaters develop, and what does that mean for things like partner specificity. And so she ties in a lot of these concepts in the context of the broader idea of these are all symbioses that can illustrate these concepts. So that's one of the things I think she does very well in this.
And again, it takes you back to concepts,
Mm-hmm.
because no one ever used to, I mean, I'm admittedly entropically more aged, and when I was first learning about symbiosis, no one ever talked about that issue. And now everyone knows about it, and that's the good thing. And again, that's why these things have to come in and get republished quite often.
Mm-hmm.
New ideas. It must be just exhausting to keep up with all that. I mean, look at the breadth of material covered in this book.
Yeah.
Ants, well, you obviously expect, since that's her interest, her love, to deal with insect microbial symbioses. But, I'm looking at fungi, I'm looking at ectorrhiza, I'm looking at all kinds of different things.
Mm-hmm.
Gene transfer, I mean, it's all, you have to know a little bit about all of it.
Mm-hmm.
And so my suspicion is this is a great capstone course for biology majors.
Yeah, that's kind of the way that I see it too. I see it as being, you will draw on what you have learned before to try to kind of come up with some synthesis of these broader topics in terms of trying to understand how do these symbiotic relationships form.
Yeah, I mean, one way to think about it is, how are they being transmitted to the next generation?
Mm-hmm.
Those associations, resource partitioning, and adaptation. These are kind of the three lenses through which to look at things. Mm-hmm. And again, I think that that's what you see here. I'm so pleased to see data throughout the book, in fact,
yeah.
as well as some lovely line drawings. It sounds like I'm doing a book review, and I guess we sort of are.
Well, we are, yeah. And I mean, there were useful things in here that you're kind of thinking about, like in terms of when she starts in chapter 2, she's talking about evolutionary origins and fates. And she mentions, for instance, addiction. So amelioration and addiction are two things that she kind of talks about as being kind of, you know, looking at evolutionary transitions between antagonistic and symbiotic relationships. So that's one of the concepts that she kind of comes at, which is the idea that for two partners to enter into a symbiotic relationship, what is their evolutionary background? Does it feed into them being more likely to form a mutualism, since that's what we're kind of defining, she's using to define symbiosis here. So would you go from being parasitic or pathogenic and moving to being more of a mutualist? So she spends a good part of chapter 2 kind of examining that idea that, you know, these, you know, is this shift from antagonism to mutualism a common one? Or is it just the way we're used to thinking about it, and so we're missing other opportunities, other contexts in which these symbioses arise?
You know, it's to me, it's interesting because years ago, I was having a conversation with Margaret McFall and and we were talking about, oh, this is what happens when I'm low on coffee. Now, this is one of those really, the protist cat fecal material, pregnant women stay away from it. What is the name of our, so you're having trouble too, so I feel much. We both drink too much, that's the problem.
Yeah, I know exactly what you're talking about, and I cannot think of the name of it. It's terrible.
How embarrassing, right? But the point that she made is that if you look in some human populations, it looks as if 80% of the population has been exposed to this.
Toxoplasma.
Toxo, that's right. So, and she said to me, if 80% of the population has been exposed to toxo, can you really call it a parasite? Can you really call it a disease? And it all comes back to context, right? And I don't have an answer to it. It reminds me very much about some recent reviews I saw about the definition of the term pathogen and host, and how we tend to use these terms, and then it kind of, you probably heard me babble on about centrisms. Look at how introductory biology is taught. It's all eukaryocentric, it's all oxycentric, and then even when we teach genetics, microbial genetics, it's coli-centric. And that really does limit the way we look at things. And then I have people come back and say, so what do? Just throw your arms out and say everything's possible? And delightful though that is to me, no student enjoys that.
No, that's true.
So I think that setting the stage on how you define things becomes important.
It does, yes.
And that's why the course you're teaching is important to me. This will be the second year that I teach this Symbiosis course as a freshman writing seminar, and I have told them I will never do that again. After my second, I get to turn it into a capstone course for my seniors in the major.
Oh, I see. Okay.
Which is where it belongs. And because no matter where you are in biology, there's a way to make this relevant.
Absolutely. I think that's true.
You tell me you like whales, we can talk about whale lice, can't we? You know what I mean? Seriously. And in a similar way, there are ways to look at it at a simplest level you want or a more complex level. So this would be an absolutely integral book for the professor.
Yeah, yes, that's the way I view it for the course that I'm going to be teaching in the fall too.
The late Ed Ledbetter, who was one of my professors at Woods Hole, miss him much. When we talked about microbiology textbooks, he was very zen with me. He says, you need 3. I said, excuse me? He goes, well, there's 1 for the student and then 2 for you. And there's some truth to that. Yeah. Because if you were to assign this book, I think well, I can't assign it to freshmen.
No.
You might be able to assign this to seniors.
I'd be reluctant to assign it to seniors, to be honest.
But at the same time, what a wonderful resource. Bits and pieces of this I'm going to be using with my freshmen. Yeah, absolutely. Absolutely. And may I also make a plug at this point? Many of the ideas that you're seeing here appear in very succinct blog form. Ed Yong, Carl Zimmer, wonderful, wonderful places to get all that done.
Yeah, absolutely. There are some very, very strong writers who are covering topics on symbiosis, topics in microbiology specifically. And that was one of the things that's the other thing that I was thinking about for the course and then in reading this book, which is I am a microbiologist, so my tendency is going to be to teach a symbiosis course featuring microbes, and this book is written very microbially based. And I wonder, like, are we neglecting some? I mean, are we neglecting systems that are not microbially based? Like you get, and maybe this comes back to definitions, because you have concepts like brood parasitism with birds. Where you have like cowbirds that will, lay an egg in another bird's nest, knock out one of their eggs, lay an egg, and then leave, and then the sucker bird, gets chomped and basically raises this cowbird young as its own to the detriment of its own offspring. And again, it's a definition thing. So that's a brief interaction, but it has an impact on the long-term viability of that species of bird who gets parasitized.
And the coevolution between them in order for the cowbird to have picked up whatever cues.
Yeah, precisely. So that's kind of one of the places where I feel like the definitions have to become a little porous, because you have these really interesting examples. Like I saw a paper a couple years ago, and I think this is the same idea of brood parasitism, but it's cuckoo birds and fairy wrens, I think it was. And so the fairy wren young, when they hatch, I think the way it works is that they have a particular song that they can sing that's like a key for the parent. And so the parent can now, so that evolved as a response to being the nest being parasitized by the cuckoo birds. And so now the fairy wren young can sing these complicated songs. I think I'm getting this right. That's a key for the parent to be able to say, oh, I feed you, but I'm not going to feed you because you can't sing this, so you must not be my offspring. And so it's an illustration of like the evolutionary arms race of like, and it's not a long-term persistent interaction, but it seems like those two species are kind of evolving in lockstep with each other because of this brood parasitism.
Despite the fact, you can talk about resource partitioning, obviously, during feeding.
Sure.
But if you this isn't this intimate association that we're used to. And it's a very interesting example, because even though they're separate from one another, look at that Darwinowing.
Yeah, I mean, it's having a significant impact on the evolution of the species if they're revamping the way that they do their own song.
Yes.
In order to try to avoid it. That's a significant impact. I mean, whether or not it's the symbiosis, that's obviously debatable, but it's a concept that ties in with that. But I think it's worth covering in a class like this. I think to leave something like that out would you want to kind of make sure you kind of shade around the edges too.
Well, I think that the danger with it, if you want to go to pedagogy, is that just like genetics, if you get sufficiently broad, then some critique can be made there's not sufficient depth. True. And it's a very difficult conundrum. I mean, there are fashions in education, like everything else, where they'll say that you don't. In fact, I just saw this thing in Slate that says details in science aren't important, concepts are, and I kind of cringed and like what? But I understand where they try to be coming from, where they're trying to come from. I understand that. I just don't agree in the sense like you can't learn language without vocabulary. So at the same time, what must you cover? And that's what you're talking about here. When you teach a course in symbiosis, what must you cover? Now, to me, I'm with stupid. I'm wearing a Vexed Muddler shirt today, right? To me, microbiology is pretty much everything, right? But I'm very cautious when I teach courses like this to cover kind of the charismatic vertebrates about things. And what works well with me is to have students pick things that interest them, and then we learn together. How many students in your course do you think?
I think I've got 17.
Oh, there you're perfect.
Good size. Yeah.
So you can all learn together, which is great. There might be a person who wants to say how RNA viruses adapt, in their associations in competition with one another. Another person who wants to look at. I had a student who wanted to talk about the Candiru. Do you know about the Candiru? That's the fish that swim through the urethra, supposedly, of human beings. Oh, okay. So,
you always get somebody who's like, you're like, yeah, do you really? Okay, all right.
And with symbiosis in particular, the number one thing that seems to get people going, and it's not in this book very much, is is the idea of behavioral modification.
Mm-hmm.
Really, that's what we talked about with cowbirds a couple of seconds ago.
Right.
But there are very odd things out there, and some wonderful video resources and things of that nature.
Yeah, yeah, definitely. That's very true. I think, yeah, there's no lack of information, that's for sure.
So this certainly is one book that's extremely valuable to you as an instructor
Mm-hmm.
when you're teaching this, especially if you're kind of, for lack of a better term, microbiocentric, as you and I are.
Right.
But what I thought was interesting, again, about this book, despite what I was just saying about it's kind of hard to cover things in detail, I mean, I'm looking right here at a connection between the way rhizobia form their associations and some of the micro, right?
Yeah.
And that's a wonderful, there's a common pathway, which I used to work in this field and I didn't know. So there you go. There's lots of stuff there. And I think that must be very exciting when you're preparing this kind of book, but it must be just exhausting to figure, what have I missed?
Oh, sure. Yeah, that's very true. That's very true. Was there, as we were kind of looking through this, there's, I thought, I don't want to skip over everything to the end, but I did want to mention that I really liked the discussion about kind of looking at, it's in the chapter titled The Success of Symbiosis, which I think is chapter 5.
Yes.
And there's some discussion about kind of looking at how symbiosis kind of intersects with a lot of different issues that we're thinking about now, things like climate change, and how humans can harness symbioses for applications like, you know, if you know that a certain insect pest is dependent upon a symbiont, can you in fact develop some kind of intervention targeted towards that symbiont in order to do pest control? And so those are interesting ideas in terms of taking these broad concepts of how symbioses evolve and are maintained, and thinking about, like, well, practically, what does this mean? And one of the things that I thought was interesting was where she starts to talk about, and as a specific example, she uses coral bleaching.
Yes.
So she's talking about symbioses in the context of climate change. And she mentions, you know, the Anthropocene, which is the idea that we are living in an era in which human activity is causing massive changes that will have an impact on ecosystems and species. And so she says, well, let's look at this idea of coral bleaching. And the idea, there's competing hypotheses about whether or not symbioses are more or less susceptible to detrimental effects from climate change because they're in partnerships. And that was an interesting discussion. So she kind of presents some evidence that, you know, maybe that's not true. It's really just system dependent. But it was interesting to, go ahead, sorry.
I just saw something about lichens getting a greater range if they're able to incorporate new partners.
Mm.
Is that an Ed Young popular article?
Sorry, I don't remember. Yeah, that one missed me.
I think that needs to be up on I'm creating work for you here. I'm sorry about that, but that needs to be up here too, because that's certainly very relevant. It could be that by changing the partnerships, we change the functionality. And this also takes you to Seth Bordenstein's idea of kind of the holobiome, for lack of a better term. And oh boy, do you get in trouble when you start talking about meta-organism versus holo-organism or whatever.
Sure.
You get, again, terminology.
Yeah.
And I do restrict myself from saying big N, little n from Jonathan Swift, but that's kind of where we're going with this, the language that we use. And I'm not smart enough to start, issuing nomenclature. But I do think that we are a community, all of us, and we tend in ecology to see things as a community. We don't think about it happens on the organismal level, and it does. Mm-hmm. There are lots of cases of particular ecosystems that are necessary for, there's the wood mouse that can't digest creosote unless it has particular microbiota associated with it.
Right.
And so if something had happened to remove that, then those mice would have to change their dietary habits or die. So this whole idea of looking at organisms as a larger whole of many other things that can shift and change with time is pretty important. So that's why the coral bleaching story was an important one.
Yeah, and I think it sets up as a broader topic of investigation. It sets up the interesting idea of looking at kind of, she's talked a bit about the evolutionary history of an organism that then enters into a symbiotic partnership. But I think another interesting question that she discusses a bit is looking, kind of projecting forward to say, if you're looking at different types of symbioses or different species involved in symbioses, is there something that makes some of them more robust to change in the partners than others? And the thing that I think is really interesting about this is that you could, if off the top of your head, you could think like, for instance, with insect endosymbionts, you might think like, oh, well, those are very tied to an insect and a symbiont, an endosymbiont partner are very kind of tied together, and they're probably very dependent on each other, and that's probably not going to change. But even within that very strict partnership, there's examples of secondary symbionts being acquired. So there's an aphid that has a Buchnera endosymbiont, but that Buchnera has lost some of the important pathways, and now it also has a secondary endosymbiont called Serratia symbiotica that supplements what's been lost from Buchnera. And so there's a couple systems like that where people are thinking and asking like, what is this? Is it an acquisition of another symbiont and those two partners will now be in balance in kind of a triad? Or is one actually going to start to replace the other? And then you go from a single symbiont system to a dual symbiont system to a single symbiont system. Like that, to me, that's another interesting question, is kind of where is it going?
And the danger is this idea, it used to be this idea of the evolutionary ladder, which is wrong. There is no evolutionary ladder. But it does kind of feed into the way that we would like to think of things. Simple, complex, which is not how reality is, but it's what we like to believe. So in a similar way, the idea that you have peripheral and then integral symbioses. Now maybe that's true, but we all tend to think it biases in that direction, and yet we can start sussing out exceptions to any rule we come up with. Now, as I recall, Laura, are there not examples of microbes, and I'm going to make this up, where one tends to overproduce tryptophan and another requires it, and then they flock up? I want to spell that, F-L-O-C. Thank you very much. They flock up. Is that not the case?
That's
there are things like.
I mean, there's certain. I mean, with tryptophan. I think that sounds familiar, but I know that there's that feed that. Yeah. Do you mean with insect endosymbionts or just in general?
No, I mean in the lab, so you can of create. You can create an association, and I'll bet you can. I haven't tried it.
Yeah, no, I have read a paper, and it's long enough ago that I'm not going to remember any of the details, but it was a lab that was looking at the formation of consortia in terms of kind of having that type of feeding going on.
Let's see, then you have something like a kefir grain, where they're doing that locally.
Right.
And I wouldn't be at all surprised. But I think that this idea of how easily or with great difficulty an association is formed is a pretty critical question.
Yeah.
And as always, we like to be reductionist and take it back to its simplest argument to start with.
Yeah, I mean, and I think that's the thing with one of the things she discusses is looking for kind of, are there genetic pathways that you can use to kind of predict like the likelihood of an organism being able to form a symbiotic partnership?
And that's kind of my prejudice about alpha proteobacters that you heard me talk about before. They're kind of preset for it.
Right. Yeah, you can make that. Yeah, you can make that argument. But I think I think this is, in terms of talking about broader concepts, you've got. This is again like another, as a lot of things are, another great example of trying to understand the balance between chance and historical contingency, right? So, you know, at some, like with the system that I studied in my postdoc, it's a endosymbiont called Lochmania that is in a relationship, a symbiotic relationship with carpenter ants, along with some other ants as well. So it's an ant tribe, but most of the ones that we worked with in the lab were carpenter ants. And so when we talked to people about them, we usually said carpenter ants because people could recognize that. But at some point, you know, you kind of think that that was likely, just like the first book we read in the book club, the John Archibald's book, they make the argument that, you know, the origin of the mitochondria was a singular event. So, you know, in this case with the Blackmania, it's thought that, you know, is it possible that it was just like a singular infection event? And so what was it in those circumstances at that time? Was it simply chance, or was it a historical contingency that led to that, or probably a combination of both? And then can you even with enough information develop some type of predictive model? You know, I think those are interesting evolutionary questions just on their own. But symbiotic systems definitely provide a good forum for trying to kind of get at those types of questions.
And I mean, you know, if nothing else, I find that students love to hear about, and this is, it is not mentioned directly. They talk a little bit about malaria and a rotobacter strain. Is that right?
Yeah, no, I do remember running across Plasmodium.
Yeah, and they do a little bit of Wolbachia here, and those are really great examples of how manipulating a symbiosis can be a benefit. And I mean, the one that I love talking about with students is where they infect mosquitoes with Wolbachia to prevent their ability to transmit dengue.
Right.
And it makes a lot of students quite nervous because, you know, the Wolbachia is transmissible and so on and so forth. But I always think of almost every association as being kind of like Thunderdome to Darwin's evolution. And so when you have two different strains of Wolbachia in the same cytoplasm, how are they doing things, right? And you talk to Seth Bordenstein, you know, he's the OG on this, right? There's very little he doesn't know about it. And it's a very interesting question to me. There are bacteriophages inside the cytoplasm too, right? You go back to a lot of protists, and you find that there are microbes that live inside different compartments of paramecium. They live in the macronucleus, the micronucleus. You know, these are fascinating subjects. And so the idea that it all comes back to is how these things associate and what each partner benefits or doesn't.
Yeah.
Very hard to believe that there's a lack of negative or positive when one organism associates with another. I have real trouble with that.
Well, and in the early stages of reading this book, I actually tweeted that out in a little series because I just wanted to poll people, because I just said, okay, like commensalism, what do we think? And I got great replies from people who essentially, they just said, I don't know, I don't really think that that's in its technical definition, where you have one partner who benefits and one partner who just simply can't be bothered at all. You know, I just don't think that's feasible.
I think a lot of this, it can be laid at the feet, honestly, of Richard Dawkins with the selfish gene. You know, the idea that everything's about fighting for resources, and so when we have, quote, DNA that doesn't do anything, there must be a cost for having that. And, you know, sometimes they get all vitalist with you and say, well, we simply don't understand the levels at which selection are working, which is great if you're in church and not so great if you're a scientist. So you have to come up with what to test. And I mean, what I like about using microbial systems to study symbiotic questions is that you can manipulate one side. And in cases of Drosophila, you can manipulate both sides.
Right.
And it is absolutely true in those kinds of associations is that both sides can really, really kind of fine-tune what the other is doing in terms of development, in terms of hormonal responses, in terms of providing an advantage against other organisms that live in that environment. So I think that that's going to be where we're going to get the kind of ideas to take to other systems is from that. And it may well be, as Professor Douglas points out, ways that will, in learning about this, that we get real benefits to humankind.
Yeah, yeah. And I'm really glad she included that, because as you've said, the scope of this is really extraordinary and impressive. I would be curious what her writing process is like, because I'm sure it is not a simple task to amass all of this, interpret it, synthesize it, and bring it together in this way. And I like that she included, I mean, this is from 2010, and so she really was thinking about things that we're still pursuing now, that are going to be at the forefront of symbiosis research for some time. And she got them in there in a really accessible way, I think. I thought that was some of that's probably some of what I am going to have. If I lift stuff directly from the book for the class, that's probably the sections that I'll have students read, is things about like the climate change section, human health, things like that. Because it helps them understand what, practically, besides just the theoretical side of things in the biology, practically what can we do with this?
And one of the really good things you can do with your class, and I've done this a little bit with my freshmen, is you can show them an excerpt of that book.
Mm-hmm.
And then you can show them what's been done since 2010.
Yeah, true. That's a really good idea.
How things have really taken off from there, and that she's really quite prophetic in a lot of ways.
Yeah, and I was thinking about that when there was a couple of sections where it was talking about the impact of symbiosis on speciation. And I mean, that's a great way to, that's a good intro to reading Seth's paper, where they were looking at, the Nasonia. So, yeah, so that's a very good idea. So I'm curious, for you in your class, so you said this is going to be your second time teaching it as a freshman writing seminar, and then you're going to advance it to a capstone course.
Yes.
What do you think you might change in that? Because that's a pretty significant transition. What do you think you might change in that?
Well, what I hope to do, because currently what the goal of the freshman course I teach, teach is a strong word, what do I preside over, is for them to learn how to read science-related material. Because a lot of times they haven't read a scientific paper.
In the freshman course or the?
In the freshman course.
Yeah, absolutely.
So what I find really useful, and this is where I am so grateful to people like Carl Zimmer and Ed Young, because they synthesize really complicated papers in an accessible way. And I often have them read a Carl Zimmer or an Ed Young article associated with, usually there's an editorial article on the paper in question, and then there's the paper.
Right.
So we start with going through those, what is missed, what was important, can you find the important points from the one essay in the actual paper? And this has been very useful to them. But what I find works the best with them is, and evaluating, of course, what's relevant and what's not, is they pick something that interests them.
Mm-hmm.
And so this is the hard part for me, is I have to work with each of them. And I don't really have a problem with that. And I tell them, this is like when a friend sets you up on a date, you got to pick the date. Your friend can't, right?
Yeah.
In a similar way, I find that they get interested in what they want to talk about. And I think the part is they would try and find out from me how much I knew about a given topic. And they would try to shy away from things I knew a lot about. And so you have to kind of play that game a little bit. And
Yeah.
I find with the biology students that I get at University of Puget Sound, especially the seniors, they are ready for something like this. So I will be able to actually go through things like definitions and examples. What I would plan to do in a capstone course is go over a simple and then a more complicated example. I would probably deal with definitions,
Mm-hmm.
current problems, that kind of thing. Keep in mind that Professor Douglas's book is 6 chapters long.
Mm-hmm.
So if you cover just aspects of each chapter for a lecture, you're not very far into the semester at all. And that enables them to learn kind of the roadmap, as it were, and then can kind of work together on what's being presented. And I find students really like that. You just have to be, again, I don't mean to tell you your business, but you just have to be very upfront with what you want them to do.
Yeah.
And what I usually do is I give a presentation myself. And that way they're able to model what they do based on what I did.
Mm-hmm.
And that seems to be helpful.
Yeah, and I think my plan is to kind of start with tackling definitions.
Yeah.
Which is what I do in my non-majors course on microbiology. The first day is like when they walk in and they, we go over the syllabus, and then one of the first things I have them do is write, what is your definition of a microbe? So like, what is a microbe? Write it down. And then on the first day, I just throw pictures at them of microbes, and I kind of say, this is Thiomargarita. If you can see it with your naked eye, that's interesting. And then at the end of the day, at the end of the lecture, I ask them if they want to revise their definition of what a microbe is. And usually they do, and they're like, okay, that's really interesting. So
very interesting, Laura, to ask them to define things in one word and create word clouds.
Okay, sure.
And do it like at the beginning of the course and the end. It's really interesting because, again, it comes back to the words we use.
Mm-hmm.
So it's fascinating.
And so I think starting with definitions in a symbiosis course, like she does in the book, she starts with definitions. I think it's important because anything they read, they're going to run across these terms.
Yeah.
And so it's good to get them to recognize from the beginning that although there are some broadly accepted definitions of those terms, some people might be using them slightly differently than others, so that they can just kind of keep in mind as they're reading, like, what does this fit in with? Does it fit with my definition? Could I argue for or against including it in my definition? Because I think that helps them, that hopefully helps them kind of engage a little more with how, if they've got a framework for how they're going to try to approach it, and then they try to fit things into it, they can either revise or defend, or, you know, it's a good exercise.
And that framework's very important. And it takes, again, back to this idea of the terminology that we use, and it does tend to shape the way that we think. And you know, when I say that, it sounds like I'm saying we should propagandize, and I've been accused of that a few times, being microbially centric as I am, and I say mea culpa, you know, I do that, but it's also the truth. But the thing is that you have to give them a framework to work through.
Mm-hmm.
I also find students really appreciate it when you talk about kind of the human aspects of it. And what really amuses them most of all is the nerd fights that take place over things. And if I'm very lucky, what'll happen is they'll kind of sneer a little bit at scientists fighting over terminology, and then I'll catch them doing it myself.
Yes.
Over that they feel strongly about. And I said, that's because it's a human profession.
Yes. Yep.
I think that the teaching of symbiosis is really exciting and really relevant, not simply because of all the changes nowadays in terms of, for example, the microbiome, our next big topic apparently,
mm-hmm.
but also because it's a swiftly moving field. People come in with kind of preconceived notions on what symbiosis is. We can fix that, and we can give them new tools for looking at things. It also, I think, philosophically is things, without going too without going too Lynn Margulis on you, I think that having like the idea of everything together is a very pleasing image to many people.
Mm-hmm.
It brings things together in a very nice way. Often biology is taught in a dispersive fashion. And one of the things that I don't care for is this big bio, little bio dichotomy that most departments have. I can't speak for yours. And you know, I really don't like the expression, you know, big bio, little bio. Students use it all the time. They're modeling behavior they see our colleagues make. And I tell them, we're all using the same tools. You can be a person interested in whales and study DNA. You can, you know, it's all biology.
Yeah.
And I think that in a very, very large department with dozens and dozens of faculty, that kind of thing might be okay. But in a smaller department, and that's what I think a strength of smaller departments is, you get to look at the whole. And that's a good place for courses like genetics and symbiosis, because they bring so many different ideas together.
Yeah, absolutely. Absolutely. Yep.
And sorry for the speech.
No, I think that's great. Yeah. And so I think we're just about at an hour.
Yeah.
But thanks again for talking with me about this book and suggesting it, because I really enjoyed it. And I know that I have wanted to read it for a while. And it's one of those things where I'd see it on my Amazon thing, and I'd be like, oh, I should really get that and read it. I don't think I have time.
Martin, yeah, he'll ask me. I understand.
Yeah, and so it was a great. I'm really glad that I read it, and it will be an important resource for me as I'm teaching symbiosis in the fall. And so we are going to do our third book club in August, and it's going to be a shorter book because we're starting to move into the semester at that point. So I think people will be getting busier. Or everybody's busy now, but maybe people will probably be getting even busier then. But we'll be doing a book by Rob Knight called Follow Your Gut. which I think might have been based on his TED Talk.
Yes.
And so hopefully we can attract a good crowd for that, because it's going to be about the human microbiome. And I
so hope that he would be here for this, that he would be there for that one, but he won't. I know that.
No, you don't think we can get him to show up for his own book?
No. But do you know my joke about Rob Knight? If you ever want something sequenced, you just go outdoors at midnight and you just say the name of your system, and the next day you get the FASTA files. That's my theory.
Do you get them from Rob Knight or Jack Gilbert, or do they compete?
Well, I don't know if compete's the right word. I mean, Rob Knight owns the planet, and then Rob Knight owns the planet, and Jack Gilbert travels the planet. How's that?
Okay. There's a symbiosis in there.
There you go. It brings it all full circle.
Well done. Okay, well, do you have any final thoughts about the book before we skedaddle?
I think that it's really easy to look at any book like this and say, I wish X had been covered or Y had been covered, and forget the amount of work and sheer breadth of what is covered. If I were to sit down and take on a task like that, you know, I think I would just shrink into a corner. So this is a fabulous book. It is well worth everyone's time. And it sounds like I'm trying to sell the book. In essence, I am, because I think it's a critical concept, but it is very much worth your time, as poor Laura had to hear from me a lot. But here it is. It's a good book, and I appreciated this conversation very much.
Yeah, me too. Me too. Thank you very much, Mark.
All right.
So we will see you the next time for Follow Your Gut.
Okay.
In August.
Bye-bye.